Welcome to Teach Yourself About Sleep. This video is the third in a series of videos on the topic of sleep. Measurement of sleep. Sleep can be characterized behaviorally by looking at the subject, but a more comprehensive and accurate assessment can be obtained by using actual measurements. The golden standard for defining sleep is from measurement of brain cortical activity using EEG, where emergence of typical waveforms indicates the appearance of sleep. In species where the central nervous system is not developed enough for EEG recordings, behavioral measures are used to define sleep. Electroencephalography is a measurement of brain electric activity in humans measured outside the bony skull. Activity of neurons creates low-voltage electric activity around neuronal networks. When activity of thousands of neurons is summated and amplified, the activity can be recorded also outside the skull. Electric activity can be assessed at different structural levels in the brain. Here is the activity of single neurons, where one spike is one action potential representing the electric activity of the neuron. An individual neuron produces an action potential occasionally and is silent the rest of the time. However, in between the activity, there are states of membrane hyperpolarization when the occurrence of action potential is not possible. These are called down states. At the level of neuronal networks in sleeping brain, the down states are synchronized so that all neurons reach the down state simultaneously. The synchronized activity is summated to a large amplitude wave in the EEG recording, which is the slow wave or delta wave. In human EEG recordings, we can observe three main states: waking, non-REM, and REM. Non-REM is further divided to three states: S1, S2, and S3, describing the depth of sleep as defined by the proportion of slow waves in the recording. Waking is characterized by low amplitude, high frequency electric activity, while in non-REM, the frequency of the wave is lower and amplitude higher. The state is characterized by large slow waves, called also delta waves. In REM sleep, the EEG looks identical to that of waking, high frequency, low amplitude waves. We can also observe specific EEG features like K-complexes and sleep spindles. These appear in specific sleep stages and are used to aid in defining the state. The number of electrodes placed on skull surface varies, reaching more than 100 in high-density EEG recordings. Using several electrodes, we can observe differences in activity in different parts of the brain. EEG recording is one part of human polysomnography measurement that includes, in addition, muscle tone recording EMG, eye movement detection EOG, heart rate and breathing measurements, and often also snoring recording. From the EEG recording, we can construct a hypnogram where the different vigilant states are presented as a function of time. Hypnogram can be described as a choreography of sleep. From this choreography, we can see that slow wave sleep, particularly the deep as 3 sleep, is slept in the beginning of the sleep period and REM sleep in the end. Sleep proceeds always in the same order. We begin from the high vigilance state of waking and go to light sleep phases S1, S2, and then to deep sleep S3, and from there to REM sleep, which again is a state of high cortical activity. One entity that includes all sleep states is called a sleep cycle, and in humans it is about 90 minutes. The EEG signal consists of a combination of waves with several frequencies that are associated with different vigilance states. These ranges represent human brain activity, but vary somewhat between different species. Slow wave sleep is characterized by low frequency from 0.5 to 4 Hz delta waves. These frequency ranges are used also in the power analysis. By construction of a power spectrum, we can extract more information from an EEG recording. The EEG recording is subjected to a mathematical procedure called fast Fourier analysis, which separates the different frequencies in the recording and constructs a frequency spectrum. The procedure decomposes the EEG signal into predefined frequency components. Here you can see the original EEG recording, of which we first construct hypnogram with separation of different vigilance states. The Fourier transformation is executed in different vigilance states separately. Here, waking, non-REM, and REM. You can see large power in delta range in non-REM sleep and theta range in REM sleep. Behavioral analysis is used when it is not possible to obtain EEG recording, for instance in small species with not advanced central nervous system. Immobility becomes then an important criterion for sleep. However, it must be emphasized that immobility is not the same as sleep. Actigraph is a movement detector that records acceleration. Basically, it only tells whether the limb where it's attached to is moving or not. An algorithm then counts, making assumptions from the degree of immobility, how much sleep is in the record. The movement algorithm is often assisted by data from temperature, heart rate, and illumination measurements from detectors that have been attached to the movement detector. A good algorithm can detect sleep rather reliably, but a true assessment of sleep can be obtained only from an EEG recording. Any algorithm must be thoroughly validated by comparing its ability to detect sleep against the golden standard for sleep assessment, which is EEG. Running wheel is an excellent movement detector for animals that are willing to use it. When the animal is on wheel, each revolution of the wheel is recorded and interpreted as waking. Infrared beam breaking can be used for activity detection, for instance in fruit flies. Here you can see the fly in a recording chamber. Infrared beam is led through the chamber and each crossing of the beam is recorded. Sensory threshold measurement is one of the classical methods to verify sleep, and it is regarded as one of the most reliable means to assess vigilance state. During sleep, the threshold to respond to stimuli increases. However, a strong stimulus is able to penetrate through to the brain and induce a reaction. In experiments, the stimulus strength is increased till reaction appears. In waking, much lower stimuli than in sleep are able to induce a reaction. This is a system to assess sensory threshold in zebrafish larvae. Here is a camera for recording the water pumps and the swimming chamber, and here you can see scenery through the camera. The stimulus is led to the chamber, and the reaction, the startle reflex, is recorded. The startle reflex consists of two phases, short latency turn and long latency turn. Several larvae can be stimulated simultaneously and counted how many of them respond to the stimulus. After sleep deprivation, fewer larvae respond. Sleep homeostasis means that the brain has a mechanism to keep count of how long we have been awake and adjust the time in sleep accordingly. This is one of the core features of sleep. The most common way to assess sleep homeostasis is sleep deprivation, meaning that the organism is kept awake for longer periods than normally. To induce prolonged wakefulness, it is very important to make sure that the stimulus used does not induce stress in the species that is studied. That is why a natural stimulus for each species must be found. Humans stay gladly awake when watching movies or playing games. Rodents are curious and inspect new objects introduced to their cages, and fish have a natural tendency to swim against water flow. After the period of sleep deprivation, sleep is recorded, and the extra sleep, as compared to normal amount of sleep, is the recovery sleep. The more you learn about sleep, the more you appreciate it. So, sleep well.